My name is Chris Thorogood and I'm the Deputy Director and the Head of Science here at the University of Oxford Botanic Garden. And today I'm going to be talking about some of the collections-based research that we do here. Specifically, I'm going to talk about some of these botanical oddities, the carnivorous plants, parasitic plants, aeroids. I'm going to talk about an interdisciplinary programme that we've established here at Oxford Botanic Garden on biomimetics. And I'm going to finish my talk with what I call astonishing plants, those that have the power to engage audiences at a time when that's really crucial. Well, let's start with a bang. This slide shows some of the carnivorous Nepenthes pitcher plants that grow on Borneo's Mount Kinabalu, a place once described by the botanist Corner as having the richest and most remarkable assemblages of plants in the world. At Oxford Botanic Garden and Arboretum, we're interested in the evolution of these extraordinary plants. How did these elaborate and beautiful traps come to be? If we first consider the left of this slide, at the top you can see the stalked digestive gland of a Drosera leaf, and next to it a photograph of the leaf with an insect ensnared on those sticky tentacles. And then below that you can just see the glands on the inside of a Nepenthes pitcher. Now for a moment, if we consider that plant in the middle, that's a mutant leaf of a non-carnivorous plant called a codium, and you can see the extension of a tendril-like structure, and at the bottom a cup-like structure, and this is broadly similar to the cup-like pitcher of a Nepenthes pitcher. It's something we call an epiacidiate leaf. What we consider has happened over the course of evolution is that a glandular leaf, rather like that of the sundew on the left, has undergone fusion of the leaf margins, leading to the production of a tubular so-called epiacidiate leaf, like we can see happening in that mutant in the middle. It probably took place via a similar spontaneous mutation that we see from time to time in plants. And what were the selection pressures driving this evolution? Now let's consider the diagram on the right. And this is based on a theory first put forward by Givnish, and it states that there is an interplay between the investment of soil-borne nutrients that we see in most plants. Consider that blue line there on the right, in which nutrients are taken up from the roots, and then prey-borne nutrients from the red broken line. You can see insects caught in the trap, and this can be invested either in photosynthesis by the photosynthetically efficient part, the lamina at the top, or the trapping efficiency of prey by the photosynthetically inefficient pitcher trap. You can see on the photograph there that the pitcher is a sort of yellowish colour. It's a tube. It's not very good at photosynthesis in order to be good at attracting insects. And in summary, it pays to invest in these photosynthetically inefficient traps in environments where there's plenty of light anyway, and the nutrients in the soil are scarce, but you can supplement those by eating insects. And this is what has given these plants a competitive edge over the other plants that they face in such places. But not all of them feed on insect prey. Several have diverged, at least partially, from the carnivorous syndrome, and they've evolved mutualistic relationships with animal partners. Our colleague Ulrika Bauer, who you can see in the photo on the right, she took that photograph of a tree shrew sitting on a Nepenthes pitcher. And the species that you can see in the diagram on the left, they all produce sturdy pitchers that derive nutrients not from insects, but from mammalian feces, these little animals that scamper onto the pitchers and defecate into them, giving them an instant dollop of nutrition in the form of manure. So it's clear that not all Nepenthes pitchers are the simple pitfall traps that all work in the same way as we once thought, sitting waiting for various insects to just slide in. And working with Professor Alain Gorelli here in Oxford and our colleagues, we've been exploring the potential for prey to behave differently on different peristome shapes. Our student Tom Strube created this graph, and here you can see plotted 3 different peristome types at different levels of wetness. And I should say at this point that wetness is key for the peristome becoming slippery and leading the insects to slide in. Now the lines show the evolution of the relative area in which prey can remain stable on the peristome. If you start at the top right, the peristome is fully dry, and here in 97% of the peristome area, insects can scuttle around without sliding off. But as surface wetness increases, the stability decreases, i.e. the chances of an insect falling in off the peristome gets higher. Now this might be expected, but it isn't the same across peristome types. As you can see following the lines from right to left, a quick decrease in the area that remains stable is observed for the green and the blue examples, but less so for the orange. And we're interested to know whether different peristome shapes and sizes and geometries leading to different prey behaviours might correlate with different prey spectra. In other words, is this astonishing assortment of picture geometries that we see in the genus Nepenthes linked to different modes of nutrition that we're yet to understand? Now I'd like to move on to a different plant group, parasitic plants. They include the world's largest flowers, economically important pests, and ecosystem engineers. All of them possess a special organ called a haustorium, which is like a plug that attaches it into its so-called host plant, penetrates their tissues, and draws from them water and food. So it forms this sort of living physiological bridge between the two plants. Many of them have abandoned photosynthesis altogether. These are called holoparasites, and they're completely dependent on other plants for their existence. The most extreme of these are called the endoparasites, and they spend their entire life cycle actually within the tissues of other plants, and they only emerge briefly to flower and set seed. And of the 4,700 or so parasitic plants described, there are 4 convergent endoparasitic lineages with about 80 species and 8 genera. And you can see those 4 independent origins mapped onto the flowering plant family tree on the left. They exist for most of their lives as a hidden endophyte, which is a series of filaments or masses of cells from which sinkers, which we can think of as being analogous to roots, tap into the conducting tissues of their host plants' roots and stems. And these plants include the largest flower on Earth, Rafflesia, that you can see in the picture on the left, a curious cream-coloured thing called Mitrostemon in the centre, Cytinus, that red and yellow one on the right, and Pilostyles, the one at the top. Working with Charles Davis from Harvard, who I'm delighted to say is giving a talk in this symposium, and other scientists, we recently examined the life history, anatomy, and molecular genetics across these 4 independent lineages. And what we saw was a high level of convergence across these clades, but also a striking trans-kingdom convergence in life history among these endoparasitic plants, but also with disparate lineages of fungi. And this is both at the molecular and the physiological level. I'll explain what I mean. Here you can see a simplified life cycle of an endoparasitic plant at the top, and beneath it that of a biotrophic fungus. And we've highlighted the key phases of the life history and how they're convergent. So starting from left to right, we see germination in response to host-derived chemical cues, followed by the production of an organ for host plant penetration and nutrient abstraction, then growth within the host tissues, and finally the production of fruiting bodies, which lead to the dispersal of numerous tiny seeds. And this intimate association between either plants growing inside other plants, or fungi inside plants, or indeed other fungi, has led to an elevation of information exchange between host and parasite. So horizontal gene transfers, the rare exchanges of genetic materials between non-mating organisms, this has been shown to be astonishingly rampant in the parasitic plants, particularly the mitochondrial genome, as shown by Charles Davis's group. And as shown in some other weedy parasites, these connections also facilitate the bidirectional movement of things like viruses, proteins, and RNAs. Now, these plants are very difficult to grow, which does make researching them difficult, and our knowledge of them to date has been very much skewed towards the impressive, better-known examples such as Rafflesia. I'll now talk about a different group of parasitic plants. This collection of illustrations shows all of the British species of Orobanche. These are root parasites. Like the examples we've seen, they lack chlorophyll. And this is a group of plants that we have a collections focus on here at Oxford to support their conservation and research. Our earlier work demonstrated that host specificity, i.e. which plants these parasites feed from, can trigger the evolution of new species in Orobanche. So we examined the population genetic structure of a plant called Orobanche minor, and we found evidence of host-specific, genetically defined clades. You can see that in the tree in the middle. Then we grew the plants in the laboratory on various hosts, and we examined what happens beneath the soil surface. And we found distinct incompatibility interactions at different developmental checkpoints. So the parasite could reach a certain point of development and then become kicked out by its host. And the overall number of compatible versus incompatible interactions at the cellular level helps determine the host specificity of the parasite. And at the population level, this can lead to genetic divergence on different hosts. They become separated and ultimately evolve to become new species. And understanding these processes can inform how we record the plants, make sense of their distributions, and in some cases inform their conservation. We recently described a new variety of Orobanche minor that's specific to brachyglottis shrubs, which are often planted in retail car parks such as IKEA. So natural populations of Orobanche have shifted onto hosts in man-made environments, and they're in the early process of evolving new species, or incipient speciation. We can also grow these plants. This is something we've been experimenting with here at Oxford Botanic Garden. Mark Brent and Russell Beaton here at the garden, our horticulturists, have successfully managed to grow some of these plants, like the one in the middle there, Orobanche picridis, growing on a potted host plant. And so together with Fred Rumsey, our collaborator from the Natural History Museum, you can see on the right, we're exploring how we can grow more of these plants that are often neglected from cultivation in gardens and from conservation efforts due to their perceived intractability to propagation. These spectacular parasites are relatives of Orobanche. I joined Ori Fragman Sapir, the Director of Science at Jerusalem Botanic Gardens, who's also giving a talk at this symposium, I'm pleased to say, to examine these plants growing out in the deserts of Israel and Palestine. These plants are traded widely for herbal medicine or have historical local importance as food. But despite their importance, little or nothing is known about the biology of most species, and their taxonomy remains confused, and that can hinder identification. Now, a growing body of research into the cultivation of some species has enabled regional supply of traditional herbal medicine at a low-level cost and intervention. In the context of a global desertification crisis, which we face, there's also the significant potential to expand cultivation of Sistanchi outside of China. We can grow it as an ancillary crop against vegetation planted to halt land degradation. It can grow on those shrubs as a parasite. But to realise this potential and to monitor trade to control unsustainable harvesting of wild populations, we need a robust taxonomy that's informed by morphological and molecular data. And so that's what I'm working on together with Julie Hawkins at Reading and our student Majed and others. We've been examining the taxonomic complexity of this genus with a view to informing how we use Sistanchi and in the context of global challenges. Another curiosity, the family Balanophораceae, a family replete with bizarre-looking parasitic plants. The exact evolutionary origins of the Balanophораceae were unclear until the introduction of molecular DNA sequence data. Now, they're seldom encountered or collected or even seen by people in most cases, and they preserve very poorly in herbaria. We really don't know a lot about most of them. On the left, you can see an example called Langsdorffia, which is a poorly known genus that looks more like deep-sea creatures than it does flowering plants. And there are just 4 species known, and they occur across Central and South America, Madagascar, and Papua New Guinea. On the right, you can see that strange potato-like structure. That's a tuber of Balanophora tobirica, and that's one that we're actually growing here at Oxford. It's not flowered yet, but we do hope that it will in the future. An important group of parasitic plants I haven't talked about yet is the mycoheterotrophs. About 10% of plant species use carbon from a fungal partner, a mycorrhizal association, at some stage in their life cycle. But the dependency on fungal carbon across the plant kingdom is variable and often unclear. On the left, you can see some plants in the genus Oxygyna, a rare and poorly known genus and then to the right of it a related genus called Thismia. And all of these plants are very poorly known to science. Many of them have been seen only once, and certainly none of them exist in cultivation. They really are some of the most mysterious of all flowering plants. You can see just how extraordinary they are. Here are some examples from Malaysia. And recently, together with Siti Munirah from the Forest Research Institute in Malaysia and Dome Nikong, a rainforest explorer, we described a new species from Gunung Sarut, which is a mountain located in the state of Terengganu in Malaysia. Now this plant, and I'm looking at the orange one, the cutout there on the slide, this has been seen twice only and may already be extinct because all attempts to relocate it have failed, and so we've given it a conservation status of critically endangered. But we know nothing about the plant's ecology or biology, and described only months ago, it may already be extinct. I'm going to move on to a project that examines the reproductive biology of aeroids in the Mediterranean. These unusual looking plants, which will be familiar to the gardeners among us, are not pollinated by bees. Rather, they dupe flies into pollinating them through mimicry, for example, of rotting flesh or dung, without offering any reward to their pollinator in the form of nectar. And together with my colleagues James McCullough, James Wickens and Luciana Carvalho from the Department of Chemistry, we've been examining the chemical signature of some of the smells that these peculiar plants produce. And interestingly, some of them mimic carrion, others mimic animal dung, and one even produces a pleasing fragrance that's attractive to bees, rather like many flowers that we're familiar with, even though its ancestors would have been unpleasant smelling, which is curious. So we're establishing a new platform with a technology called TDGCMS and a sample collection methodology using the plants growing here at the Botanic Garden to test the hypothesis that these different aromas are more closely linked to the resources that they're mimicking chemically, dung or carrion for example, than has been previously considered. And interestingly, we do see differences in the traces produced by the dung mimics and the carrion mimics. So you can see the ones in red at the top mimic carrion, and the three black ones at the bottom mimic dung, and they do have compounds that split these two groups apart. And so we're now extending this methodology across the various collection of aeroids that we grow here at the garden. A final area of research we do here at the garden linked to our collections is biomimetics, an interdisciplinary field in which principles of engineering and biology are applied to the synthesis of systems that mimic biological processes. Together with my colleagues James Guan and Finn Box, we examined the slippery surfaces of the Nepenthes pitcher that I discussed earlier. Inspired by the plant, we created artificial surfaces to explore the capability of trapping, retaining and directing liquid droplets. And our work revealed a potential mechanism for developing systems in which the transport of droplets is controlled by so-called energy railings, those grooves that you can see on the image on the right of this slide. These railings provide a biomimetic means of transporting and sorting droplets, and they could be used in droplet-based fluidic devices. It could enable the efficient mass transport of liquids along predetermined pathways, inkjet printers for example. And these observations also offer insights into the evolution of the plants. They show that the pitfall trapping mechanism is enhanced by the water-infused grooves on the slippery rim, driving prey into the trap in a way that's much more tightly controlled than we considered previously. Finally, decreasing environmental awareness and disconnection with nature, these are symptoms of a growing problem that's been described as nature deficit disorder. And plants in particular are established to be a blind spot in the human psyche that needs that extra layer of focus. Although we owe our very existence to plants, unlike animals, many of us scarcely even notice them, and this has been described metaphorically as something called plant blindness. 2 decades since that phenomenon was described, plant blindness is more relevant than it's ever been. For example, in the context of an increasing rate of biodiversity loss and extinction. In fact, 2 in 5 of the world's plant species are threatened with extinction globally. And it won't be possible to address this alarming trend unless we can foster a greater environmental care and awareness of the importance of plants. So I'm going to talk briefly about how some of the strange and unusual plants that I've examined in this talk can help us to do that. Here are some examples of plants that we know people find inspiring by sharing them with our online audiences at Oxford Botanic Garden and Arboretum. And in particular, the botanical oddities, the curiosities that we've examined in this talk, they can be very effective at this. They can break down people's perceptions about plants, how they are and how they behave, and bring new people into the conversation about the importance of plants. In fact, these examples of unusual plants that you can see here on this slide, these reached tens of millions of people around the world, stopping people in their tracks to think about plants. Maybe examples such as these can help foster a greater care and appreciation for plants at a time when that's needed very badly. Because in light of the significant global challenges we face, botanic gardens need to be agile and inquisitive in finding new approaches to communication that engage people with the wonder and the importance of plants.